Hi, everybody. It's me. It's Dr. Kestler from Lorain County Community College, and welcome to today's COVID-video. I'm going to start by reminding people to wear masks. I believe that it's making a difference, and that's an important thing to do. So I'm going to take mine off right now, but I just want to remind you I have a new mask, as you can see, and just remember to use the mask, practice social distancing. I think we are starting to see some change, but it is something that you have to continue with. Don't give up. Don't stop, and don't say, "Yay, we're all good," and go back to exactly normal. I think that was a mistake we made a month ago, and we're not going to make that same mistake again. First of all, I'll tell you my temperature today is 95.9 in Fahrenheit, which is 35.5 in centigrade. I want to spend some time talking about vaccines. And, I have spent, before I start talking about vaccines, I just want to show you something here, a little bit of data from the pandemic. And that is I'm showing you that, I titled this, but I'm unhappy with the way I titled this slide. I said the shifting target of SARS-CoV-2 really is that makes it sound like the virus is doing this. No, it's you. Your behavior, your actions are helping this happen. So it's not that the virus is doing this, it's you. You're the ones in control here. You are very much in control. Right now, as you can see, it looks like the United States, Brazil, even South Africa are appearing better, and I think the real battleground for the virus, to get rid of the virus, will be India going forward from here on. So now I want to talk a little bit about vaccines, and the COVID vaccines are promising. And of course, I've got I cannot begin a vaccine talk without talking about these two guys, Edward Jenner and Don Henderson. Jenner, of course, 250 years ago, discovers the first live attenuated vaccine. That live attenuated vaccine was for smallpox. And the guy on the right, Don Henderson, grew up in Lakewood, Ohio, went to Oberlin College in Ohio, and went to a place called the University of Rochester for medical school. And I understand they have a very fine program up there. And he worked for the Centers for Disease Control, and he ran the WHO's program on smallpox. So these two individuals are the bookends of smallpox. You have the beginning of the vaccination campaign and the end. Now, before I go any farther about talking about successful COVID vaccines, which I'm going to, there's some very promising results. But before I do that, I want to just remind you something. First of all of us need to sit back, take a breath, relax, and try to understand each other's position. You can spend a lot of time arguing and calling other people idiots, and that's going to get no one anywhere. Try talking to somebody about their feelings about vaccine. Let me say this. It is perfectly appropriate to be afraid of vaccination. It is perfectly appropriate. I'm not going to tell you how to feel. I'm going to tell you that if you have concerns, that's good. That means you're a person. You're a good person. You're thinking about it. There. Now I've said that. So before we go out there and call each other all jerks about vaccines, let me tell you this. I'm going to tell you this is the facts. Vaccine campaigns have been successful despite having vaccinated every single individual. There is no such thing as universal vaccination. Universal vaccination, the Easter Bunny, Santa Claus, the Tooth Fairy, all don't exist. Oops, I shouldn't have let all that stuff go. Whoops, my bad. Santa does exist. I just want to point that out. Santa is real. But all the rest of those things are not. There's no such thing as universal vaccination. We've never been able to universally vaccinate people. We can achieve herd immunity without universal vaccination. All we got to do is get enough people who are either immunized naturally through getting the disease, or by vaccine to have enough immunity to stop the vaccine in its tracks. And there's some data that I'm not going to talk about this today, but it might be quite a low number to control this particular one. So Henderson, one of the things that he came up with is case-based or spot vaccination. And the idea was that if you have an outbreak, you go into a particular area, because you're never going to be able to vaccinate every person in the world to be able to achieve a vaccine effect. You just can't do it. So that's where I'm going to start. We can do this, even if not everybody does. Nobody's going to come to your house with a gun, knock down the door, and say, "Get your vaccine." That has never happened in my lifetime, and I'm pretty old, so I'm guessing that it will not happen in the future. So that's all. Having said that, and all, and we can now move on. First of all, I'm going to talk about two vaccines today. The two vaccines are the Moderna vaccine. This one had a paper out last week. I mentioned it in the video last week, but I want to show you a little bit about the data of it. It is very encouraging. Small number of people were involved in this vaccine study, but this is more or less a safety study. The Oxford and the Moderna study excuse me, spent an awful lot of time looking at side effects of vaccines. That is obviously something we're always concerned of. We have a drug or whatever, we look at side effects. We want to know what are the bad things that are going to happen. So they extensively spent a lot of time. Pain at the site of injection was a frequently indicated one. In the Oxford study, they actually introduced a Tylenol arm of the vaccine studies where they would give people Tylenol to lessen that amount of pain. I personally would rather not do that right away. I would do it after initial development of symptoms from the vaccine. You want to get a little sick from the vaccine because that's going to make a better effect. That is going to give you a better effect. So let's go on. Let me tell you about the Moderna vaccine. Really clever, a brand new kind of vaccine. Both of these are novel vaccines. And what they did was they took an RNA molecule and they stitched in the gene. Mostly they were genes that were involved with the outer envelope protein of the SARS-CoV-2 virus. And that would hopefully make one would make a very nice immune response to that. And they did seem to get some nice immunity. And in this picture here, you can see antibodies to various proteins, to a couple of proteins that are on the virus. One is an epitope that is directly binds to the receptor on the host, and another one is just a general antibody. And then they look at, first of all, at day one, there's not very many antibodies, IgG antibodies available. Then by depending on the dose, by day 36, you definitely have an immune response in there. And I think if you go to the higher dose. In this Moderna study, they dropped the idea of a higher dose of 250 micrograms because of more side effects. So they decided that it was good enough to go with the 100. So that's one of the reasons for this study, is to try to figure things out. On the very, very right-hand side of both of the panels A and B, you can see the presence of convalescent antibodies. So they're measuring it from people who actually got SARS, got sick, and made an immune response. And so they're measuring the vaccine-induced responses versus natural responses. And they do seem to be quite robust. It looks like we're doing a good job with that particular vaccine. On the bottom, these are assays that are designed to look for something called neutralization, virus neutralization. And virus neutralization is when you completely stop the virus in its track. And these are various kinds of assays that should go towards that. One looking panel D directly at the virus, and panel C looking at a surrogate marker for the virus. That brings us to the now one of the things that happens when we get to the Oxford study. The Oxford study is much bigger. It's about 10 times the size of number of people who were enrolled. They really spent a lot of time looking at side effects, and they actually included Tylenol in their study. So they were giving some people Tylenol, and then they got rid of some of these side effects in there. Very interesting kind of a vaccine because it basically is an adenovirus of chimpanzees. So we're going to a whole new species, and it's an adenovirus that they had been working on for a different disease, MERS, coronavirus, same family as SARS-CoV-2. And so they were working on a vaccine in chimpanzees, and they tried it this way. They took the adenovirus, they spliced in the spike protein, the outer membrane protein, into that virus, and that is the basis of this whole story. And so then you basically give somebody this, or you inject some of this into the people, and we see what we get from this particular story. And this is one of the things that is called a common cold. So that's the kind of thing they're looking at. Much bigger study, and you can see here are some of the side effects that they were looking at. And they also included in their study a totally unrelated vaccine. They included a meningococcal vaccine for meningitis disease, bacterial meningitis. And so that's their control, which is a pretty good control because everybody's getting vaccinated in this, but some are getting vaccinated to something totally unrelated to SARS-CoV-2, to coronavirus. And then what they came up with is, I'll just show you a couple of data here. First of all, these are ELISA studies where we're looking at antibodies. We're looking at IgG. And you can see that the people who got the meningitis vaccine, adenovirus vaccine, got nothing. They had no antibody response, any measurable titers. These are log scales, so it goes up by a factor of 10. They have very little antibodies that protect against SARS-CoV-2. If you look at the one dose versus one dose plus a boost, you can see that there is a significant amount of antibodies all the way out 56 days out, which is what is that, 2 months out. We still see neutralizing antibodies, and then they compare, same as in the Moderna study, they're comparing on the very right-hand side there. They're looking at the convalescent samples. Last thing I want to look at in this data is is a neutralization assay, and you can see they've got the meningococcal data in light blue, and you can see that basically after about 35 days, you can see that there is with this vaccine, there is neutralizing antibodies that are comparable to the neutralizing antibodies found in convalescent plasma. And this is really great news. For those of you who do not want to partake, we got you. We're going to protect you. The herd will protect the ones who don't want to participate. That's fine. So nobody's going to put a gun to your head and tell you have to get a vaccine. That is not going to happen. All right. Last thing I want to talk about is some more good news. And this good news is about, this was a study out about dexamethasone therapy in people who are severely ill. I just want to point out the data here, and it shows you that when you give dexamethasone therapy, it's definitely beneficial. In the case of people who are on mechanical ventilation, in the case of people who receive oxygen therapy, it is beneficial. So the severe parts of the disease, it is not necessarily benefiting all patients. So this is not something that you should say, well, damn, I have COVID. I should go out and I should um, um, make a vaccine. I should take the dexamethasone. No, that is not what you should do at all. Because it's only for the people who are extremely sick. All right, I'm going to stop that right there. That's our report this week, and I think that we have some good stuff to be cheerful about, and we should be cheerful. We should be. There is a light at the end of the tunnel. I can see on the horizon that we're going to move beyond this, and that is that's fabulous news. It doesn't mean that there's not going to be some more troubles. I think we're going to see, even if we see fewer cases, we're going to see still a high mortality rate going into the next few weeks. But at least the leading edge of the storm seems to maybe have passed. All right. Remember to be good to each other, and may you all persist like a lentivirus. Thank you, and have a good day.